Life cycle. Schistosomes have indirect digentic life cycle involving sexual reproduction in vertebrate-dependent host and asexual reproduction in the snail intermediate host. Parasites are transmitted between hosts by motile aquatic stages, which actively seek hosts. Female worms produce numerous eggs, around 200 to 3,000 per day, which seek to exit the host by penetrating the gut or bladder wall and being passed with the host feces or urine. When deposited in water, the embryonated eggs hatch, releasing free-swimming merostidia, which only live for several hours. In that time, they actively seek suitable intermediate hosts or snails. All Schistosoma species demonstrate host specificity for particular snails. Schistosoma mansoni infects Bamphilarea species. Schistosoma japonicum infects Oncomelina species, while Schistosoma hematobium infects Balanus species. The Bericidia invade the soft tissues of the snail and form a mother sporocyst near the site of penetration. Daughter sporocysts are produced 2 to 6 weeks after infection and then migrate to the other organs in the snail. Schistosomes do not produce radiate stages. Instead, the sporocysts produce cercaria, which are released in the water in their thousand beginning 4 weeks after infection. The fork-tailed cercaria are rapid swimmers, and they vertically swim to the surface of the water and then sink to the bottom for up to 3 days. They are attracted to skin secretions, and when they come into contact with the prospective definitive host, they attach and actively penetrate the skin within minutes, losing their tails in the process. Inside the host, the schistosomula little schistosomes are carried in the blood or lymph to the portal veins in the liver, where they develop for 3 weeks. Young worms then bear and migrate to their predilection sites in the veins of the gut or the bladder. Egg production begins from 4 to 8 weeks after infection, and adult worms normally live for 2 to 5 years, although some may survive much longer. This is the life cycle of Schistosoma. As you see, cercaria released by the snail into water and free swimming penetrate the skin of the man. This is a cercaria loose tail during penetration and becomes schistosomula through the circulation, migrate to the portal blood in the liver and mature into adults. As you see here, the female in the third adult worm migrate to the mesenteric venules of bowel or rectum, laying eggs and circulate to the liver and kidney stools, venous plexus of bladder. As you see, Schistosoma hematobium, Schistosoma japonicum, Schistosoma mansoni. The egg hatch releasing merostidia according to the different species of mollusks or snail species, according to different species of Schistosoma. Then the sporocyst in the snail, through the successive generation, they produce cercaria. Cercaria is in infective stage, and as you see, the eggs are the diagnostic stages. Pathology and symptoms. Symptoms of schistosomiasis are caused by double secretion or reaction, to the worm eggs. Not all eggs laid by female worms successfully penetrate the gut or bladder walls. May are swept away in the circulation and become trapped in organs, where they elicit strong granulomatous response. استجابة حبيبية لهذه الأنسجة. Eggs become surrounded by inflammatory cells forming characteristic pseudotubercles, zygoranate, which may coalesce to form large granulomatous reactions, polyps. Encapsulated eggs die and eventually calcify. بتموت البيوض وبتتكلس. Intestinal schistosomiasis can result in abdominal pain, diarrhea, a blood in stool. Liver enlargement is common in advanced cases and frequently associated with accumulation of fluid in peritoneal cavity and hypertension of the abdominal blood vessels. In such cases, the There may also be enlargement of spleen. هذه الأعراض تحدث نتيجة وجود الشيستوسوما في الانتستين. Classical sign of urogenital schistosomiasis is hematuria, blood in urine. طبعا نزول الدم مع البول. Fibrosis of bladder and urethra, kidney damage, or sometimes diagnosed in advanced cases. Bladder cancer is another possible complication in the later stages. In women, urogenital schistosomiasis may present with genital lesions, vaginal bleeding, pain during sexual intercourse, and nodules in the vulva. While in men, urogenital schistosomiasis can produce pathology of seminal vesicles, prostates, and other organs. This disease may also have other long-term irreversible consequences, including infertility. Catamaya fever, another primary condition called catamaya fever, may also develop from the infection with these worms, and it can be very difficult to recognize. Symptoms include fever with severe itching, urticaria, rash, liver and spleen enlargement, and bronchospasms. تشنج القصبات الهوائية. Differential diagnosis infections are conventionally diagnosed by detection of fluke eggs in the fecal or urine samples, often after concentration or by sedimentation, flotation, or filtration techniques. The eggs are sufficiently characteristic to facilitate specific diagnosis. On occasion, microscopy of rectal biopsies have been used to diagnose Schistosoma hematobium infections. Immunoserological tests have been developed to detect host antibodies against infection, but they have experienced cross-reactivity problems and cannot discriminate between previous and active infection. More recently, molecular techniques have been used to detect parasite antigens or DNA in host samples. Treatment and control. The drug of choice for treatment of all Schistosoma species is brezoguantil, a single oral dose being very effective, with low toxicity and good tolerance. But snail population may be reduced by strategic use of molluscicides, nicotinamide or copper sulfate, draining marshes and swamps, clearing channels of vegetation. Irrigation practices can be modified to avoid long-standing still waters, and different or improved crops can be used which are less dependent or lengthily immersion in water. In endemic areas, farmers and visitors need to be aware of dangers of emerging potentially contaminated waters.